Okay, let's move on to the second section, using protein data bank. You perhaps learned about or heard about a name called PDB, which stands for Protein Data Bank. This PDB is the database for protein structures. It's the only database and unified database that contains all this protein structure information. It serves through different mirrors, such as American PDB and RCSB, or European PDB from WWPDB. You can select one of those and access to the structural data, which is shared across different mirror websites. And then what we are going to learn in this lecture is about how to interpret and use the structure information that are deposited in PDB. Here's the PDB statistics as of the beginning of this year. In RCSB, there were more than 210K structures deposited in PDB, where crystal structures compose about 85% of the entire structural database, followed by EM and NMR structures. And here's the growth over every year. So you can see that crystal structure has been grown most rapidly and still steadily growing, whereas EM structure has been growing really rapidly in the last 10 years. What we're gonna learn today is how to understand the biology from PDB, and followed by how to interpret PDB by knowing the biochemical experiments that were used for determining structures. And then in 3 and 4, we are going to learn how to extract information of protein dynamics and atomic details in PDB with some cautions when you interpret the structure database. Okay, let's start with the first part, understanding biology from PDB. So why do we have this huge structural database? There's some unique feature that you can understand about biology by looking at their structures. This is called structural biology. And then the unique aspect you can learn about biology from atomic details of proteins made were the main reason for establishing this PDB database. If you look at left-hand side, you can see the structures of proteins like virus capsids. You can see that they are very tight and has some thick shell that nothing can actually interrupt inside. If you look at tiny bit of these antibodies, you can see that the shape of grasping part that you can understand this will roll as police inside our body. And you can also see this GRO-EL and GRO-ES that it'll rotate as machine in order to do some kind of protein function. If you look at filament, you can see that they are quite well packed and tight shaped so that it can act as some structured proteins in our body. So by looking at these protein structures and shapes, you can understand their functions and roles in our body. Protein carries their roles by what is called biological unit, which is usually quaternary structure. If you look at this hemoglobin, it's actually made of 4 chains. And then single chain does not mean anything. The hemoglobin only folds when they have 4 units, copy of 4 units. And then by having 4 units, you can get much more efficient oxygen carriage. So we call this what the protein actually that exists in our biology as biological units. And then this has different repository shown here. It's not on PDB, but in other sites that are only containing biological units of proteins structures. The reason why there's a different definitions of biological units and repository is because what is deposited in PDB is not actually biological unit. It's asymmetric units. As I showed in the previous slide, that the majority of protein structures are determined by crystallography. And then crystallography is defined by asymmetric units because it has symmetry definitions, as we learned in the structural determination lecture. And then these asymmetric units often doesn't correspond to biological units. For example, in this case, asymmetric unit is actually 4 copies of this protein. But then real biological unit is just single chain. So what you observe in crystal packing is definitely different from what exists in protein or in biology. That's why there is a distinction between biounits and asymmetric units. To help you understand better, there are many cases when asymmetric units doesn't match the biological unit. For hemoglobin, people determined their structures in many different ways. Some of them are determined only containing 2, some of them determined into 4, and then some are determined with the 8 chains. So in this case, this PDB, the asymmetric unit is made of 2 units, 2 asymmetric units. And in this PDB, it's built of 4 asymmetric units. And this one, 8. But all are just hemoglobins. And regardless of what the asymmetric unit was, the biounits are the same. It should be 4 chains. So asymmetric units is something that represent the experimental condition, whereas biounits are what represent the biological condition. So there's a difference there. And this is extremely important because when you look at protein structure from PDB, especially when it's a crystal structure, you have to extract biounits from asymmetric unit. And then because of that, people determine the structures based on asymmetric units, but annotate what biounits should be. And this is definitely done by the authors of that experimental structure, but often it's not correct. Because here, converting from asymmetric unit into biounits, it's followed by some subjective interpretation. If you carefully look at crystal structure of this protein here, then which one do you think would be the biological unit? It's hard to judge. So authors thought maybe this is the biological unit, or this one can be, or even the author thought it may not form any oligomer, and it'll work as a single monomer. So people studied whether authors' annotations are correct in terms of thermodynamics, and then argued that about 7% of author annotations of biounits are wrong. And this is extremely important when you interpret the structure from PDB, especially when it's a crystal structure. You often forget that the asymmetric unit you're looking at from the PDB is not actual biological units. If you open up a structure like this, it has 2 zincs coordinate to the protein, and you'll think these things are real, but that's not actually real. This one behind is real metal coordination, but this one is artifact. This can only be built when there is a crystal symmetry unit here. So this is only formed at the crystal condition and not formed in the biological condition. And then you can only know this by generating the symmetric mates from your crystal structure. And here is another example. If you open up this PDB, then you'll see ligand lying at the surface. Of course, this is not real structure that exists in biology. This ligand doesn't bind to this protein. But the reason why this ligand located here is because of the symmetric unit here. What that means is that these ligands exist only at the crystal condition and not in biological condition. So this is something you have to be really careful when you interpret crystal structures. It's really important to build the symmetric mates of your crystal structure in order to understand and interpret the correct biological structure of protein, and if that's not influenced by any asymmetric units or symmetry mates in the crystal structure. The second information you can get from PDB is the sequence that was actually used to determine the protein structure. Often this is identical to the actual protein sequence, but sometimes it is not. So you have to be sometimes careful looking at this. And then there are some residues that appear in sequence residue record, but not appears in PDB. These are called missing residues. So if you follow the sequence record part in the PDB, there should be 15 residues here, but not determined in PDB. And the reason for that is because you cannot judge where the sequence protein backbone should lie based on the experimental data. Previously we learned that the way how we get the protein structure is based on the electron density converted from diffraction data. But then often there are cases when electron density is too blurry that you cannot follow where the sequence should go. In that case, just experimentalists leave that part alone and only annotate the backbones of proteins where they think is reliable. In that case, there should be missing chain for 15 residues locating here. But no worries, this does not mean protein is broken there. It's just that undetermined, and the loop part will be flopping around this region. There are also inserted residues. And then if you look at PDB, you can see sometimes like this, the residue number has ABC. And then this is called inserted residues. I'm not going to go details, but these are often happens in some protein systems where insertions and deletions are quite common, such as antibodies. These are regular sequences that exist in sequence record. And it's just numbering is different from other proteins. At this part, we'll learn the biochemistry influenced in PDB. When you read the PDB, you have to be really careful on biochemical experiments that was used to determine the structure. Because structure determination is a very hard job. Experimentalists introduce many different biochemical modifications in order to build crystals or purify their structures or tag what proteins it is and so on. And then this kind of modification to protein sequence or structures definitely influence the protein structure. But then biochemists try best not to change the original structure, but you have to interpret what these modifications are when you're using it. Here I'm showing you a common shared modifications across different PDBs. One of well-known modification is selenomethionine. We learned about this in the crystal structure determination lecture. We introduced selenium at the methionine part in order to phase the protein crystals. And we learned that the selenium mutation isn't actually influencing much of the original methionine based protein structure. But you can easily identify from your PDB if it contains amino acid name of MSC and something very similar to methionine, but having selenium instead of sulfur. And the second modification is peptide tags. And the first part of protein structure determination is protein purification. You have to collect as many proteins as possible. And by adding these tags, you can get help on collecting more and more purified proteins. But then at the structure determination part, often these tags are attached to protein structure, and sometimes they could influence the protein structure minimally. But you have to know that these are not actual biologically meaningful residues. It's just used for purifying proteins. And then when you use the protein structure, you must delete this part because it's not part of the biological unit. And the third type of modification is domain insertion or deletions. It was extremely difficult to determine some sort of proteins. And then some scientists invented how to stabilize proteins by inserting certain domains like this. And this is purely for protein stabilization or crystallization, and no biological meaning for that. And we assume that this domain insertion doesn't affect much of the protein structure. But anyways, it's not because a part of the protein biological unit. When you use this structure, you have to delete that part. There are also domain splitting at the structure determination. This also happened quite commonly in crystallographic protein structure determination for membrane proteins. If you look at membrane proteins, they are made of extracellular domain, intracellular domain, and transmembrane domain. But it becomes extremely hard to determine entire structure in a single experiment, because these transmembrane domains have different characteristics. So what people usually did was they split the chain so that one part becomes extracellular domain, the other part becomes intracellular domain, and then they determine the structure separately by growing crystals separately. And then they are deposited in different PDB ID like this. But essentially, they belong to the same chain. So if you are gonna interpret this protein, you have to somehow build a full chain connected structure by making some linkers between this. There's also effect from solvent media. Solvent media are often used to stabilize and crystallize proteins. And then these are non-biological ligands. If you open up PDBs, often you see this DMSO or glycerols surrounding protein structure. The experimentalists deposit their entry in this way, even though they are not actual biological ligand, because the PDB should contain all the experimental details, how the structure was determined. But anyways, you have to keep in mind that these are not ligands. These are just solvents that were used for protein purification or stabilization, and you have to interpret your structure without these small molecules. Now, this is for soluble proteins. Same story holds for the membrane proteins. For membrane protein structure determination, people use what is called detergents. And detergents mimics the membrane bilayer condition. These detergents are quite hydrophobic in one side and hydrophilic in the other side. So you can stabilize the membrane protein without using actual lipid bilayer. So by using detergents, people were able to crystallize these membrane proteins, and there are many membrane protein structures determined using detergents. In some cases, detergents are not crystallized well, so they are just blurry and not deposited in PDB. But in the cases, there are some detergents still remaining in the structure, showing how detergents are surrounding these membrane proteins. But again, these are not actual biologically meaningful ligands. They are just small molecules used for determining the protein structure.